Dr. Ladd is the founder and chief medical officer of Neuroscience Technologies and Neurofluidics. He is also one of Duke's very own, serving as the director of Duke Neuro Innovations Program and professor at our medical school with a focus in functional neurosurgery. Dr. Ladd will talk to you about his path to pursuing an MD PhD in the field of neurosurgery and neuroscience, and how he's harnessed his background to build 2 successful neuroscience-based startups. Please join me in welcoming Dr. Ladd.
All right, well, thank you. Amazing job organizing this event and great turnout, so I appreciate the invitation. So Nikhil asked me to talk about our experience with neurofluidics and using neuroscience and medical innovation. The first project, we call it, or startup, was neurofluidics we were involved in. My background was here, so as an undergrad, I went to a very small school, Cal State Los Angeles in Southern California. That's where I grew up. As part of this early entrance program. So a little bit different path, but the advantage was that you got to start early. So I was close to home, but maybe a different path than many of you experience in the traditional college life. But then I made up for that in medical school and graduate school. And I think there's different opportunities, as the prior speakers have said, that will sort of change the way you do things and the way you look at things. So for me, early entrance program was one. So we skipped high school, started college early, did traditional college, and then decided, okay, what do I want to do? And I was always interested in neuroscience and was heading towards medicine. But I said I wanted to actually learn the science side of things and be a clinician scientist. So that's what I did, and did the MD PhD program in Chicago. From there, the PhD taught me many things, in addition to grantsmanship and hypothesis-driven research, a lot of late nights and multiple late nights in a row, which you get in medical school, but I think the PhD really accentuates that even more, and that trains you well for neurosurgery. So it all fits together somehow. Then I went to Stanford for residency and fellowship, and that was a fantastic, another sort of turning point and a great opportunity. And being at a place like Stanford, similar to Joe, I said, what do I want to learn while I'm here? And certainly we're there for the didactics and the clinical training and to be a great neurosurgeon, but also the other things that you can learn while you're at a great institution. And so I said I want to learn neuromodulation, which is a subspecialty in neurosurgery, which focuses on electricity in the nervous system. So how do you modulate the brain, the spinal cord, the peripheral nerves, the autonomic nervous system to try to restore or rebalance or tune the nervous system. And then the other unique program was the biodesign program, was how do you go from an idea to commercializing it. And it would be a shame being in there and not to be a part of that process. So neurofluidics was our first endeavor that basically applied the biodesign process to a problem in neuroscience that we saw in the clinics and in the ICU specifically. Subsequently to that, we took that same process, applied it to NeuroAccess since coming to Duke, and we're in the process of starting a third project that is still in stealth mode. So people talk about translational research. I think it often gets lost in translation, and you have to start with an unmet clinical need, and I think that's always important. So you have to look at who is your patient, why are you doing this, what problem are you really trying to solve, and never forget that. And that process has been left to a few individuals or to serendipity, but the things we use in the operating room or in the OR, in the clinic, ICU, someone developed, right? And so understanding how that process came about and the tool you're using in your hand, that's 5 to 10 or 20 years of someone's life that went into. So I said, as a physician, you can certainly impact patient care on a one-to-one basis. People have talked about how do you measure your life. Wade Christensen's book, a great book I agree everyone should read. What impact will you have on society? And I think as a physician, you can make a tremendous impact. I think supervising other providers, having a team, you can magnify that impact. Training other physicians, like at Duke University, in the neurosurgery training program here, you can further amplify that impact. But by developing a new solution, I think you can exponentially magnify the impact you make by changing standard of care, and many more so even if you're doing 500 to 1,000 surgeries every year for 20 years. It's 20,000 patients and their families, let's say 60,000 people. But if you change the way medicine is practiced, you can impact many, many more. So I think all of these things, this is from the biodesign slide deck, but all these things are important when you're considering how to build something. I think that each of you here have that opportunity to bring mechanical principles, materials, access method, desired tissue effect, drugs, to be that missing link and think about how can what you're doing actually benefit someone. But starting off with who that someone is, what the problem is, and then working to solve that problem. So within neurosurgery, there's many subspecialties, as we said. There's spine, there's brain tumors, there's vascular, endovascular, pediatrics, peripheral nerve, critical care, and then stereotactic functional, which is the area I chose to focus in, and also known as neuromodulation. And so I think we can go in depth in one particular area, in neuromodulation specifically, and that's enough to keep someone busy for a whole career, and then some, and stick to what you're good at. So neurofluidics, we started as a resident, so after doing a biodesign program, and the unmet need there was we saw many patients who had an aneurysmal subarachnoid hemorrhage, and this is when a blister on a blood vessel bursts and then releases a bolus of blood into the basal cisterns around the brain and the brainstem. And it's treated with aneurysm clipping and coiling, which stops the bleeding, but then you have 5, 10, 20 cc's of blood that shouldn't be there that then break down over the next 2 weeks. And so we saw patients that are otherwise young and healthy, in their mid-40s, family, kids, were devastated because no one really knows that they have an aneurysm. Sort of a ticking time bomb. When it ruptures, it's the worst headache of your life. And 50% of patients die before even making it to the hospital. And those 50% that do make it tend to have a very long, protracted course. So after seeing this again and again over neurosurgery 7 years of residency, I said, so we have to do something. And so right now, still the current standard of care is we put a tube in, drill a hole in the head, put a tube in the ventricle, which is the spinal fluid cavity, and drain the fluid to relieve pressure and also to drain some of the blood out and into a gravity bag. So I said, and there's been 50 years of data to show that blood is bad. The more blood you have, the worse your outcomes are, and the chance of delayed stroke goes up exponentially. So the need statement here was a minimally invasive platform for targeting specific pathogens in the CSF that improves outcomes and saves lives. And so as you think about these, you can certainly come up with different needs and say, I want to improve or reduce the rate of delayed strokes by 50%, or I want to reduce mortality by 1/3. So you can get as specific as you want to, but that was sort of the genesis for the work that followed. And it was several years, and I still serve as their chief medical officer. Minutronics, which is a company in Minnesota, acquired the technology a couple years ago, and now they're looking to second, third, fourth applications of it. So again, it's a platform technology. So a simple concept is, as an undergrad, as a medical student, we saw many, I was interested in neuroscience, and we said, we can pinpoint these pathogens in the CSF, but you can't really do anything about it. And why is that? Because I think it's that intersection of bioengineering and medicine and understanding the pathophysiology of the disease. So we said, well, if we know what the pathogen is, let's target it and remove it. And it sounds easy, but it's not so easy. As when you embark on any project, it's harder, longer, and more expensive than you ever had. But the idea basically was to create active circulation in the spinal fluid. Right now it's passive diffusion in the brain, choroid plexus in the center of the ventricle makes CSF, which is an ultrafiltrated plasma. It's about 20 milliliters an hour. The body reabsorbs it so that instead of 480 over the course of a day, there's about 150 milliliters in any given person. And it's sort of slow, passive diffusion. And we said, can we accelerate that process, prevent blockage of the arachnoid granulations where the CSF gets absorbed, and target diseases that are relegated to neurology, where we know the pathogen, but it's watchful waiting is our standard of care, like aneurysmal subarachnoid hemorrhage. So we developed a neurophoresis system, is what we call it, so modeled after things like plasmapheresis of the systemic blood. But CSF is a lot simpler. It's 99% water, a little bit of albumin, and then electrolytes. So you don't have to worry about platelets, you don't have to worry about clotting, you don't have to worry about a lot of things you do in the blood. So in many ways, it's a much easier system. It's just a very privileged space because things are trapped on the other side of the blood-brain barrier. And so being able to access that has been a challenge to date, and being able to manipulate that fluid has also been a deterrent. So we said, let's target the filtration to the disease of interest. And you can imagine whether it be size exclusion, which is what we started with because that was the most straightforward, but immunoaffinity, charged, hydrophobic. We thought about many different things about depending on the disease, but we wanted to, picking your first indication is also important, which we'll get into in a second. A pump and then a unique multi-lumen catheter that allows pressure and volume to be constant. When you do a spinal tap, many of you have probably heard of lumbar puncture. Spinal tap is a very common procedure done in the hospital when someone has a neurologic condition that we don't know what it is, but are worried about infection. You can take about 10 milliliters of CSF as a standard LP lumbar puncture. If you take 20, people start to develop spinal headache because the dura starts to sag, and so you're limited by pressure and volume. So the idea was, how can you minimize the amount of fluid that's outside the body at any given time, but still turn over large volumes? So not 10 milliliters, but 5 liters of CSF. So do what the body would take a month to do, but do it in 24 hours. And that's the only way to do that is by keeping pressure volume fixed so that the patient doesn't notice any change in that pressure volume curve. So that was the basic concept behind the neurophoresis system. And so we said, we want to do many things, serve as a platform technology for things that are relegated to neurology, and have it as a new neurointerventional tool within neuromodulation. And that includes removing neurotoxic factors, having targeted drug delivery. Right now, all we do in terms of drug delivery is infuse Dilaudid, morphine for pain, and baclofen for post-stroke spasticity. But I think over the next 10 years, there will be lots of applications, whether it be in gene therapy delivery or specific drugs to modulate other conditions that have a problem with bypassing the blood-brain barrier. So really conditioning the CSF and serving as a monitoring platform. So in terms of subarachnoid hemorrhage, we talked about it. This is the starburst pattern that you see on CT scans when someone has the worst headache of their life. They present with a starburst of blood that basically fills the basal cisterns. That's treated by occluding the aneurysm, whether it be through an endovascular procedure that coils and seals off that ruptured site, or by an open craniotomy that's put across the neck. But there's this bolus of blood that's sitting outside the vessel, and so a patient survives that initial insult, but then 2 weeks later, this blood leads to vasospasm and causes a delayed stroke. So they made it out of the first window, but 2 weeks later they have a second hit, and that's what we were targeting. So we've built the system, we've met with the FDA, it's an ID clinical pathway. It ends up being PMA, which is the most rigorous pathway out there. But the company that we partnered with, or that acquired Neuroscience, has the wherewithal, the funding, the passion, and the belief that it'll help advance standard of care for tens of thousands of patients, 30,000 patients every year that develop subarachnoid hemorrhage that currently we don't know what the problem is, but actually do something about it. And basically the picture here, this is what spinal fluid should look like, crystal clear. This is what it looks like in a subarachnoid hemorrhage. So, and with the animal studies we've done, we can turn this back into this. And rather than targeting specific cytokines, so people target tumor necrosis factor, or IL-6, or endothelin, different inflammatory pathways that happen downstream of the red cell. But it's all based on when a red blood cell lyses, it releases hemoglobin. Hemoglobin gets oxidized, and then over the next 2 weeks, it starts 20 or more different cascades. And people have targeted this through pharmaceutical approaches over the last 50 years. But instead of going after the branches, we said, let's go for the trunk and treat. Let's target the red cell, let's target the hemoglobin early, and then not worry about all the different branches, because if you eliminate the source of the problem, that should improve outcomes. So that's the approach we took. The reason we picked subarachnoid hemorrhage is because patients are often otherwise young and healthy. They're devastated by this, and there's a long history showing that blood is bad. The other reason is, from a design perspective, it's large molecules with short clinical endpoints. So red blood cells are about 5 microns. We can remove them very easily, and at 2 weeks, you know whether you're successful. Did they develop a stroke, or did they not? So you don't have to do a 1-year or 5-year clinical trial. You know at 30 days at the most. The FDA requires a 90-day modified Rankin score in terms of a stroke scale. But it's, from a clinical trial perspective, it's much shorter in terms of showing your efficacy. So that was also part of the decision making. So I'm excited to be a part of that. We're going into human clinical trials later this year. It's been a long road from even as a resident to a fellow to now faculty. I've been a faculty here 4 years. So I think we'll get to the 5 Ps at the end, but perseverance and passion are 2 of them, and I think that's important to making anything successful. I view it as a platform for a lot of different things, though. So subarachnoid hemorrhage is the first. The world expert in cryptococcal meningitis is the chair of infectious disease here at Duke, Dr. Perfect, and so he literally wrote the book, Cryptococcal meningitis, and so he said, you know, this is another indication. This is a fungus that doesn't belong in the spinal fluid, but patients that are either immunocompromised or organ transplant patients tend to develop it. It's about 10,000 patients a year, so not a huge market, and traditional venture-backed companies don't tackle it because it's not $1 billion type market. But you know, these are otherwise young, healthy patients that can be devastated. And in the U.S., 20% mortality, worldwide 70% mortality. So, and as a neurosurgeon, we see these patients, they languish. We can shunt. We put a tube that diverts the spinal fluid and delivers it to the belly and sort of temporizes things. But we treat them with systemic IV antifungals, and the efficacy of that, again, is not so good. So there was a recent clinical trial in the New England Journal showing that the mortality and morbidity directly correlated with the colony-forming units in the spinal fluid. So on the right here, these are the organisms. This is the capsule around it. So the organism itself is around 3 to 5 microns. The capsule can be much larger, 10 to 20 microns. Again, our endpoints are short. You know within a week or 2 whether that patient is going to survive or not. And the molecules or the pathogens are large. So from a clinical trial standpoint, very more feasible and easier to show a reduction. So in the studies we've done so far, we can reduce organism burden by 99% in 24 hours. Amphotericin B, which is the main IV systemic antibiotic, takes 2 weeks and reduces it about 70%. So that's our standard of care. So the standard of care bar is very low to cross over. So I think we can do some good. Another application, the third platform is in cooling. So in spinal cord injury patients, this was a football player that was recently treated with systemic hypothermia that made a miraculous recovery. He was quadriplegic, was treated with systemic hypothermia, and made a full recovery. We're already running out of time.
Yeah, about 2 minutes.
2 minutes, okay. So I view it as a platform for a lot of different things, and I'm excited to help bring the technology to bear to really change the way we look at a lot of these things that have been relegated to neurology and have a targeted neurosurgical treatment for them. These are some other neuroinflammatory disorders. These are more research. So the 3 I showed you, there's very clear understanding of what we're doing and the mechanism. Alzheimer's, we don't know exactly what it is, if it's A-beta or phospho-tau. So that's more research with a big R as opposed to development with a big D for the R&D. So the 5 Ps, I think, start off with a patient in mind and a clinical problem. The prior panelist said you have to be passionate about what you're doing. I agree. I mean, you have to be hungry in the morning and really be wanting to make a difference in that patient group. Perseverance. Oftentimes when things look bleak, you may be the only one there to persevere and see things through. And this is an Edison quote: many of life's failures are people who did not realize how close they were to success when they gave up. You have to build the people around you, and having a great team is critical. And then really build a good product that makes a difference and does what your initial intent was, starting with the patient. So, thanks. We're hiring 2 research aides, so if any of you are next year,